Okay, we're back.
All right, so we're gonna get started. We are starting a little bit late, but I made my presentation squishable because I know that we have another presentation coming up and then the tour. So I just want to take a quick moment and thank Zach and Sarah for putting this together. Thank you, Kevin, for finding us a lovely place to meet in. And thank you to Helen for being the first person to invite me to be part of this group.
And our generous lunch.
Yes.
Yes.
Yay. So as was mentioned, my name is Diane Pleva. I am the Plant Diagnostic Outreach Specialist at the University of Illinois Plant Clinic, which is a lot of words that doesn't mean a lot to many people. Basically what it means is I diagnose most of the plants that come through the clinic. So today I'm going to talk about some common plant pathogens found in water recycling systems. And in doing some of my lit review and looking into this, one thing that kept being mentioned over and over again is that pathogens that cause root rots and pathogens that are well adapted to aquatic environments are likely to be the most problematic, which really should not be a huge surprise, but I thought I would stress that since this is something that was, again, stressed in many of the papers that I was reading. Root rots, I think, makes sense for, this recirculating water is going to be coming into contact with the roots in some way, whether it's in a gravity bed or if it's even one of the misting beds, you're eventually going to have that water probably making its way down to the roots. And obviously we're talking about systems that have a large amount of water in them, and these pathogens that can actually not just survive in water, but are actually favored by very wet environments are going to do the best. So I'm going to spend most of my time talking about water molds. That's kind of a common term, or a common name given to them, but Pythium and Phytophthora. We've already heard a little bit about that from Shankun's presentation. There are also true fungi. So water molds are actually not true fungi. There are a group of organisms called oomycetes. I'll talk a little bit about the differences in a moment. But there are some true fungi that are often found or can be found in these recycling or recirculating systems. Phytoleuopsis and Rhizoctonia, those are both true root pathogens. So those cause root rots. Something like Fusarium, which again has already been mentioned, Fusarium, we call it a root nibbler, so it will cause some damage to the roots, but really the issue is when it moves into the vascular system of plants, and it can cause wilting, which can cause overall plant death, which is not, great for most people. I get very excited, but most of our producers are not. There are a number of viruses that can move in hydroponic systems. I only mentioned a couple here because, frankly, it seems like every week a new plant virus is found. But these are kind of important. TMV, or tobacco mosaic virus, has a huge host range, including ornamentals and vegetable crops and specialty crops. So TMV is a big problem. Tomato mosaic virus, that's going to be a huge issue on all solanaceous crops. So one thing to keep in mind when you're looking at viruses, the name it's not necessarily specific to that host. So if you see, again, something like tobacco mosaic, that does not mean it only affects tobacco. And again, in the case of TMV, it has a huge host range. There aren't too many bacteria that are usually problematic in terms of plant pathogens in hydroponic systems. Cleptobacter michiganensis causes bacterial wilt or bacterial canker, on tomato, which can cause huge losses, so that can be problematic, but generally speaking, we don't see too many bacterial issues. Nematodes, again, we don't tend to see a lot of them. Root knot nematode can circulate, as can lesion. So these are just some examples. One other thing that's very interesting to me is that fungi can sometimes actually vector these viruses. So sometimes having a fungal infection is going to act as that source of essentially of wounding that's going to allow those viruses to enter the plant. So like I said, I'm going to spend most of my time talking about water molds because that's what is most problematic in these sorts of systems. Pythium and Phytophthora are the two classic root rot water molds. As I mentioned, these are not true fungi. These are oomycetes. They're often called fungal-like organisms. And it's important to know the difference between the two. They act like fungi. They reproduce very similar to fungi. They infect similar to fungi. They grow like fungi. But they are different in a couple of very important ways. Oomycetes, their cell wall is made up of cellulose, so similar to what we see in plants, whereas true fungi, the cell wall is made of chitin, so more like the insect exoskeleton. This can make it really difficult to target oomycetes once they've begun infection of a plant, because obviously anything that's going to degrade their cellulose cell wall could potentially have some off-target effects on the plant host. There's also some different metabolic pathways that are different between those two groups. And so again, if you have a fungicide that relies on some of these metabolic pathways, they may not be effective against oomycetes. And so this is one of those reasons that people will say, well, why does it really matter if I know I have a root rot? Why is it really that critical to figure out, is it a true fungus or is it an oomycete? Depending on what management you're planning on doing, it may be really critical because you don't want to, dose a system with a fungicide if that's not going to be effective. It's just a bunch of wasted time and money. Again, these cause root rots. There are some aerial issues that can be caused, some above ground issues that can be caused by Pythium and Phytophthora, but generally speaking, we're going to be focusing on the root rot aspect. And these are favored by wet environments. So in spring, when we plant our fields outside and we have a really wet spring and farmers have to go and replant, often that's due to the seeds just washing out, but that can also be due to their damping off or stand loss. And that's usually due to these sorts of oomycete root rots. And so my point is just that these sorts of pathogens are a problem not just in hydroponic and aquaponic situations, they're also a problem to our kind of more traditional growers as well. And it's something that we see every spring. So again, those wet environments. The reason that these are so favored by the wet environments is that they have mobile spores, which is kind of unusual. But they have these mobile spores, these zoospores, that are produced from Sporangia, and if the video works, you'll get to see them move. But basically, you have these asexual structures, and these Sporangia are these. The Sporangia is the asexual structure, and the zoospores will be expelled from the sporangium, and they can actually basically swim through water, and they can swim directionally. So it's not just floating in a current; it's actually targeted swimming. They are also able to sense root exudates, and they're able, therefore, to swim toward a root, which makes them, again, very infectious when we have very wet situations where these zoospores can swim and easily start new infections. They also produce reproductive structures that are resting structures, which means that once they contaminate a field or a system, they can stick around for years. So this is one of the reasons that these particular pathogens are really problematic. And this is an example of, this is a soybean plant that I infected with a Pythium root rot. But these are kind of the classic symptoms. You'll see that this taproot is completely rotted. We have a dark brown staining. The taproot is missing. And then these little secondary roots, these side roots have come out and they are trying to establish. And at this point, it's probably a 50-50 chance whether or not this plant is going to survive. But if it does, it's probably going to be a little stunted. It may not be as productive. This was actually a semi-resistant line. So if you can imagine, we will have some Pythium either species that are so virulent, or we will have some hosts that are so susceptible that the plants will die before they emerge. Like that little radicle comes out, the Pythium basically swarms that radicle, and you end up with just a rotting seed. So again, this can be really problematic in certain situations. Okay, now comes the true test. Oh no! That was not what's supposed to happen. Don't know if I'm going to be able to play this. I've never tried playing a... Okay, well anyway. Oh! No! Come on! We all want to see you swim. Okay, I'm going to give up. Basically what you'd see here, this is the Sporangia, and these little blobs look kind of like jelly beans that are being expelled from it. These are the individual zoospores, and what's really interesting, and I'm so bummed you can't see this, because what's so beautiful about it, and it's actually, this particular video is set to music, so it's extra awesome. But what's awesome is that you see these little, you kind of get this gelatinous matrix of spores that are expelled. So you have your sporangium, and this little kind of gelatinous ooze comes out, and you have this kind of blob, and then all of a sudden it like explodes. Like all the spores just go in different directions, and they're all swimming independently. So again, they're not just being swept down a current, they're actually able to swim and target that plant exudate. And then I mentioned that they also form resting structures. These are called oospores, or oospores, if you're trying to make reading a very boring paper interesting. But these are called oospores. These are double-walled structures. These are actually sexual structures. So you can see here there's this round, it kind of looks like a sunny-side-up egg to me. But it's got like a double-walled structure. And so these are very well adapted to persisting in environments. And here we have one in a root section. You're like, okay, sure. Here we have, let's see, 1, 2, 3, 4, 5, 6, 7, 8. We have at least 8. in this very small section of root. And you can imagine this was actually not a particularly bad infestation. So you can imagine that there are roots, we were joking earlier at lunch, that you can have roots that are more Pythium than they are root. And that can become a big problem because, again, this can persist in systems. It can persist in soil, and it can persist in recycled water systems. Really quickly, a little bit about the University of Illinois Plant Clinic, if you're not familiar with us. We are the federally designated plant diagnostic laboratory in Illinois. So most states have a federally designated plant diagnostic lab. Some states have multiple, some states have none. In Illinois, there's one, and it's us. We are located at the Urbana campus. So if you're ever in Urbana and really want to see some sick plants, you are actually welcome to come by. We are an open lab. If it's in summer, I'm probably going to say like, oh, nice to see you, and then move on because we do process quite a number of plants. We are part of the National Plant Diagnostic Network. It's a federally funded network of these diagnostic labs. We cooperate with state and federal agencies. We mostly talk with people from Illinois Department of Agriculture, USDA, Illinois Department of Natural Resources, Illinois Crop Improvement. But we're basically here as a resource and as a source of either collaboration or just as a consulting resource for a number of these organizations. We obviously cooperate with other diagnostic labs across the country. We pay very close attention to what's going on, like in Purdue and in Iowa State, because we're seeing a lot of the same problems as they are. But we do try to keep in touch, and we have national meetings every couple of years. And our mission is diagnostics, training, and outreach. In terms of who we serve, most people, the agronomic producers, makes sense, right? We are in Illinois. I know that we are currently up in Chicagoland, but Illinois is known for its corn and beans, and we see a lot of corn and beans at the plant clinic. But we also see a number of nursery plants. A number of nursery producers will send plants to us either for disease issues or for phytosanitary testing. We've actually started getting a larger number of specialty crop producers, so things like hydroponics or aquaponics or things like high tunnel or hoop house. Specialty vegetables, herbs, things like that. And so we're seeing a much wider range of both hosts and also growing conditions, which is really interesting and also can be very overwhelming because we try to give recommendations to everyone, and we're trying to give recommendations sometimes in systems where there may not be a lot of good research to give us recommendations that we can feel confident in. So we try to work with our clients. We see a number of samples coming in from arborists, landscapers, lawn care companies, so a number of kind of green professionals. We do work with homeowners and private citizens. We obviously collaborate with researchers, professors, and students, and our permits allow us to receive samples from across the United States and from all overseas U.S. territories. So a couple times a year we get a cool package from Puerto Rico from an off-season nursery. They have really cool stamps, so that's always fun. We have a woman that has little children at home, so she gets to take them. And then in terms of what we provide, in 2017 we diagnosed 3,600 samples, which I personally am very proud of. We're a pretty big clinic, especially in this region, so I like to throw that number up as much as I can. We also try to provide IPM-based management recommendations for these diagnoses, and that's, again, where things can become a little bit more difficult when we're working with either unusual situations or unusual hosts. But again, we do as much research as we can so that we're giving you research-based recommendations, and we're not just saying, I don't know, you figure it out. So it's not really helpful. Most clients care how to fix the problem. They don't actually care what the problem is, right? I get excited when I see a new disease or a new pest. Most people don't care. They just want to know how to make it go away. And so that's what we try to focus on. And we also provide information to the general public via fact sheets, newsletters, presentations, blog posts, etc. And our services include pathogen, insect, and plant ID and management. We do a pretty wide number of nematode assays, so soil samples and soil testing and screening. And then we recently picked up this herbicide resistance in amaranthus species. So if any of you, again, are also familiar with traditional cropping systems and you've read about either Palmer amaranth or water hemp and the concerns there, also with herbicide resistance to those weeds, we're doing some of that testing. In terms of methods for pathogen or pest identification, we kind of have three that we use quite a bit at the clinic. The first is morphological, which is a fancy way of saying what it looks like. So what color it is, what shape it is, the size of the spores, the arrangement of the spores. Are they born in a fruiting structure? Are they just lying on the surface of a leaf? Those are all morphological characteristics, and that's traditionally what has been used to identify pathogens. So up here I've got a picture. These are Sporangia This is Pythium. So the video that did not work was more of a pear-shaped Sporangia, and that was from a Phytophthora species. Pythium tends to be a little bit more rounded, but that's a kind of a general rule of thumb. Here you can see them all lined up. I thought it was a really pretty picture. But again, this morphological, this can be very valuable. To be honest, it's fairly cheap, it's fairly quick. So that's one of the reasons that we continue to use morphological identification. However, it may or may not be enough to get down to species or subspecies or strain specificity. To be fair, again, most of our clients don't really care what specific species of Phytophthora is infecting. They just want to know how to fix it. And the good news is that for a lot of these diseases, management will be usually fairly conserved across that genus. So if it's not, then we're going to dig down and try and figure out exactly what we're working with. There are serological tests that we use. These are used for specific pathogens. So one of the nice things also about morphological is that someone brings a sample into us. We take that sample, we're going to incubate it, we may culture it as needed, and then we just give you a list of everything that we find on that sample and then how to manage those things. And there are going to be some things that we'll say, oh, we found evidence of this, however, it's a fairly minor infestation or a fairly minor infection. We do not recommend management at this time. And then we'll say this was a severe infection and it needs to be managed. So we try to also give a little bit of guidance in terms of how critical that management is. With serological tests, it's a test for a specific pathogen. And so the issue with that is that if I test something for tobacco mosaic virus and it's negative, all that tells me is that it doesn't have tobacco mosaic virus. It doesn't tell me what the problem is, right? And so that's why we try to use these serological tests a little bit more sparingly and only when we suspect particular issues. Serological, they're usually based on antibodies or proteins. They're actually very similar to pregnancy tests. So you take your plant tissue, you grind it up, you stick this strip in it, and the plant sample matter moves up the strip. If you get one line, it's a negative, and if you get 2 lines, you're a pathogen mommy. I feel like I should start bringing chocolate cigars or something to hand out, but I get way more excited about that than most people do. But thank you for laughing, because sometimes that joke goes really badly. Serological, we're actually moving away from this type of testing, not so much because it's a fairly easy way of testing. The issue is that there are certain commercial companies that refuse to accept our serological tests because the possibility of a false positive is fairly high. And so if you're doing something like phytosanitary testing where you have to prove that your plants are free of a certain disease, they don't want to risk that they send a bunch of plants to us, we test them, we charge you, and then it turns out that they're positive. Well, are they positive or is the kit just not very specific? And so I don't mean to imply that all serological testing is not accurate. That's one of the reasons that we're exploring some other options. But we are able to test for Phytophthora, for example, with a serological test. And then molecular is going to be similar to serological in that it's a test for a specific pathogen. So again, it's a yes or a no. The nice thing with the molecular testing is that it's based on genetic material, so usually DNA in most of our cases. And that's going to be much more specific than the serological test. And with that, we're usually looking for presence or absence of pathogens in mixed samples. So again, one of the nice things is you don't have to try and isolate it. You don't have to try and get single spore cultures or anything like that. We are also using this to test, for example, for Palmer amaranth seed contaminating clean seed. So Palmer amaranth is on the noxious seed list for a number of states. It's becoming a really big issue. And so we will receive mixed seed lots in, and we can test for the presence of that one particular pest in a mixed sample. Whereas it used to be that you would go through and you would hand pick out what could potentially be a Palmer amaranth seed. They're about the size of a poppy seed. So you're hand picking out everything that's the size of a poppy seed, and then you put it in the greenhouse and hope that it grows so that you can identify it, which, as you can imagine, takes quite a while and is very frustrating. So in terms of screening water for the presence of Pythium and Phytophthora, so this is something that is really only going to be useful in these sort of recirculating water systems. Most farmers are not going to worry about this. They want to know if there's a problem in their plants, but whether or not the water, they're usually not irrigating from a specific source, so they're not too concerned. Although this, I guess, could be used for source water for irrigation as well. So I'll have to think about expanding that. Currently, we're using morphological and serological tests. And again, the benefit of that is that they're both fairly easy. They don't take too much time. They're not particularly expensive. The drawback is that we have to have infected plants. And so that means that if you, as a hydroponic or an aquaponic producer, want to check if your water is contaminated, my advice is going to be plant something and let's see what happens, which is not what a lot of people want to hear right before they invest a lot of time and money and effort into planting their entire system. So we do have a couple of producers that we kind of basically have like bait plants that we leave in their systems and see what happens, but it's not ideal. Baiting is a possibility. This is where we would receive water and then we use autoclaved, usually leaf discs. And we put that sterile plant tissue in the water. As I mentioned before, zoospores are able to target, so they can swim to plant exudate or a source of plant exudate. And then after 10, 20 days, we check and see, is there any infection occurring on that leaf disc? It's really cool when it works. The problem is that if it doesn't work, there's no good positive control. So you're not sure if it didn't work, was it because there's no pathogen present, or is it because I didn't wait long enough, or I used the wrong species of leaf sample? And that's been one of the issues, is that certain pathogens respond better to different species of bait. And so, do you test 20 species of bait to see if there are possibly 20 different, which is infecting the water? It's messy, it's complicated, and frankly, it takes just too long. So it's not really ideal. What we're hoping to move toward is this molecular testing that, again, would only require the water, so we'd only have to get a water sample. And this is something that was already discussed in terms of the filtering the water through a membrane and then doing a DNA extraction on that membrane. So hopefully what we're doing is we're capturing the DNA of anything that was in that water, be it bacterial, viral, or fungal, oomycete, whatever that was. Once that DNA is extracted from the membrane, we can run it through PCR. And what we're looking for there is we're using specific primers, and we're looking for amplification. And basically what it boils down to is if you have amplification, it indicates that that pathogen was present. And if you don't have amplification, it means that it's not. So that's what we're kind of moving towards. So currently we can test for 3, soon to be 4, Pythium species. So currently we can test for Pythium irregularis, Sylvaticum, and Ultimum. And these are 3 species that are known to cause problem in hydroponic and aquaponic situations. The Pythium infanodromatum I'm actually really excited about because that's kind of one of the big baddies in the hydroponic, aquaponic world. But I have to wait till I get my positive control before I can feel really confident in that test. So we haven't really deployed that one yet. We can also test for Phytophthora as a genus, and frankly, I would rather be able to test for Pythium as a genus as well, because again, most people don't care what species is affecting their plants, because management is going to be very much the same. The problem is that Pythium is a very varied genus, and so trying to come up with a genus-specific primer and probe has been very difficult. I've not been able to find anyone that has had success with that. And so because we have that limitation, we instead have to test for specific species. With Phytophthora, we're able to just test for the whole genus. The other reason that could be an issue with Pythium is that Pythium oleandrum, is anyone familiar? It's sometimes used as a biocontrol agent. So you'd want to make sure that we're not accidentally picking up Pythium oleandrum if we were looking at Pythium as a genus. We tested 11 water samples collected by Helen's lab for Pythium, and for these 3, the irregularis, Sylvaticum, and Ultimum, and all were negative, which is great news for the producers and really boring news for me. But that's okay. I would rather have the producers, in a good shape. And so this is just an example of visualization of those results. As I mentioned, we can test for 3 different species of Pythium. So the yellow, the red, and the green lines are those 3 different species. These are our positive controls showing amplification, so again, showing that that pathogen is present in our positive control, which is good. And then all of this noise that you're seeing that doesn't amplify, those were all of our water samples. So again, just showing that we're not getting any amplification in any of those other controls. And I promise that there are actual data values associated with these that we analyze. We don't just look at these pictures, but I thought this was a much prettier way of expressing this data than a bunch of numbers. So in terms of future directions and where I'd like to see the service go, we have a couple of steps that we need to take to make sure that we're kind of covering all of our bases. What I'd like to do next is artificially infest water with these Pythium species that we know we can test for, also artificially infest water with Phytophthora. And so that may involve either growing infected plants and then collecting the water that we use to irrigate those plants. It could also involve just putting a plug of a pure culture in a bowl of water, because again, these are water molds. They do very well in very wet situations. But basically, we're going to artificially infest this water. do the membrane collection, DNA extraction, and make sure that we are actually able to capture that DNA. We're going to check that there is a lack of inhibitors. If we have inhibition during PCR, that can lead to a false negative. However, we've already kind of worked on that, and there does not seem to be any inhibition going on. So again, all those 11 negatives, I feel pretty confident are actual negatives. I'd like to expand this Pythium species list. It's not actually that difficult to find the specific primers that I need in the literature. The hard part is finding a known positive control so that we, again, feel very confident in our testing. I'd like to try the Phytophthora genus assay. It should work really well, but I'd like to actually give that a shot. And then eventually I'd like to be able to offer this phytopathogen water screening as a standard service at the plant clinic. So again, something that if you have either a system that you're experiencing problems with, or you set up a new system and you just want to ensure that you do not have infestation of your system, you could just send us water samples as opposed to having to, plant either plant seeds or put plugs in, wait a couple of weeks, and then send those samples to us. So this is kind of where we'd like to eventually end up in, hopefully by the end of the year. That's my goal. We'll see how that goes. And then my references and acknowledgements, again, thank you so much to everyone that put this together. If you have any questions, I'm happy to answer them.
One or two quick questions for Diane before we transition.
Yeah.
What is the most common disease that you find amongst field plants and then amongst controlled environments?
So in controlled environments, in systems like what we're talking about, it's going to be a Pythium root rot. Again, the species, up until recently, we haven't gone down into species level because our clients don't care, frankly, and we didn't have the opportunity to do so. I would expect, once we get that Pythium infantodermatum, I would expect that to be a little bit more common just because that's in the literature a lot more in terms of it being problematic. In terms of outdoor plants, that's a much broader category. We see a lot of foliar issues, and I know it's really broad, but the samples that we see at the clinic are everything from a blue spruce tree to a cucurbit plant. So when we're talking about that wide of a variety, it's hard to pinpoint specific pathogens, but a lot of foliar pathogens, and true fungi are going to be our most common group of pathogens. So I joke that, if someone ever walks up to you and is like, 5 minutes to save the world, what type of pathogen is infecting this plant? Just go with fungus, because that's your most likely bet. But again, in these sorts of situations where we have very wet environments, and that's true for whether it be a controlled environment or if it's a field situation where we have saturated soils and very wet conditions, we expect to see these types of oomycete root rots very, very frequently. And they're ubiquitous in the soil environment, so it's not a surprise to find them anywhere across the state. Yes.
Within the recirculating environments like aquaponics, hydroponics, are you seeing any trends in the two, aqua versus hydroponics?
No, and part of this is because when we receive samples from clients, we ask them to give us some background information, but we don't always get that background information, so sometimes it's hard for us to know, is this coming from a hydroponic system, an aquaponic system, just a traditional greenhouse or a hoop house. So that can be a little bit more difficult, and if we continue down this partnership, I'm going to try to pull that out a little bit more so I have a better data set to work with. We have one very regular client that I was actually going to use as a case study if I had to use up time, but it looks like we're getting along. But we do have a very regular client that they have a bad infestation of Pythium in their recirculating system. It's a hydroponic system, and if the issue that they're having is that when they set their system up, they did not put in any redundancy. They didn't put in any ability to turn off certain bays. And so the problem is that you have one giant system. It's heavily infested with Pythium. They're also organic, so that limits their treatment options. And their best treatment would be to shut the whole thing down and just bleach it, frankly, and then let it dry out, sanitize it, and then move on. They can't do that, though, because they're already in contracts with local restaurants to provide fresh produce, and they cannot shut down for 4 to 6 weeks. That it would take to clean the system and then restart.
Yeah.
So I can say that if I had one kind of take-home message, it would be please put in shut-off valves in your system, because that's been what we've just been running into with them, and it's been a long journey of they're treating with copper, and so we're trying to find that very fine line between what will suppress the Pythium but not cause phytotoxicity. We also, one of the things they've started doing is they're now harvesting at 4 weeks instead of at 6 weeks, because if you let those plants go to 6 weeks, they literally start melting. Like the infestation's so bad. If they can harvest at 4 weeks, they still have a harvestable product. It's a lot smaller, but so we try to work like that, but it can be difficult because again, each system is a little different and people have different constraints. But again, in terms of trending between the two, since we haven't really in the past asked people to identify, I don't have any good data on that. Yeah. Yes.
Does the plant clinic do any work with beneficial microbes or recommending beneficials to outcompete?
Yeah, so we will recommend beneficial microbes when we have data that supports their use, or when if something is labeled for use against a particular pathogen or pest issue, we will include that. So when we send final reports, the reports include what we found, if we suspect anything, and then management recommendations. And those management recommendations include cultural control, and it also includes things like beneficials or biological control. So it's not just a list of apply this, or this chemical.
Great.
In terms of do we work with them? Not as much, because we are much more focused on the pathogen side of things. But if I can get some of these positive controls, I would actually be really interested in the Pythium oligendrum and seeing if people use it, how does it circulate? There was a question earlier about whether the disinfection affected. Was that a question that you had? Yeah. And so I'd be really interested in, we could actually track. You could dose it with a certain amount. We could test the water to see at what level we're being able to recover that DNA, and then see after a round or two of disinfestation, is that level dropping? Is it maintained itself? Is it completely gone? We could actually start working with that, but currently we focus on pests and pathogens.
Right now I'm interested in identifying microbes that are in healthy systems, and then being able to escalate those to outcompete just as a...
Yeah, and that's the sort of thing that one could do with some of that 16S sequencing that was mentioned earlier. That is something that I think is in our capability. It's just not something that we usually offer as a service, because it's not something that most people, again, are going to be really focused on. But if that is something you're interested in, you can always contact us. I may put you in contact with someone else, depending on what you're looking for. But we can try and work with that. Yeah.
Cool.
Okay, one more real fast, because I know Kevin.
Just about your producer that's having problems. Do they germinate in their system or outside of their system?
They germinate in their system.
Should try to germinate.
That's one of the things that we recommended. The problem is that, to the best of my knowledge, to the best of my knowledge, the system was set up. It was set up quite a while ago when hydroponics was still kind of a new thing, and so they didn't set new thing popularly, and they didn't. I don't think they really set up a full system, and so they don't have. When I recommended this idea of germinating outside, they said they don't have the space or the capability to do that. So I think they have very limited space, and it's basically entirely taken up with their system. They finally bought a second reservoir, which is very exciting, but it's not super helpful if you can't turn off the whole system, because, as I mentioned, you have those oomycetes or those oospores that can be resting structures. They can get stuck anywhere in the system. And then, so just changing out that reservoir is not necessarily going to be that helpful. Yeah. Okay. If you have any other questions, you have our contact information on the little circles, and you're more than welcome to come up and talk to me afterwards as well. Okay, thank you.
We'll talk about 3 things today, 2 projects that I've been working on, and one of my students, Brendan, just came in here, so he might be able to help chime in on the projects. Then also I want to have more of a general discussion about maybe creating a network around aquaponics or hydroponics, both individually, separately, because as far as I know, there is no network. There isn't much connection going on in this world. And if it involves maybe just a once a year meeting or something like this, I would be more than happy to organize. So maybe we'll have just a soft discussion about that, and then we'll do a tour. We'll show you our aquaponic systems and our greenhouse, and answer any questions about our program as well. So first of all, my job is the urban agriculture coordinator. So I manage our urban ag program that involves aquaponics. It involves our greenhouse lab, and it involves a lot of places on campus where we grow food, we sell it. So it's a whole diverse operation. And I don't claim to be an expert in any one of the things that I do in this program, And so, to be honest, aquaponics is kind of a small part of my job, but I've spent some years on it, and I have some dedicated students. So there have been some things that we have moved, but I don't know if I would call it really hard research. So the two projects I want to talk about today are adding water quality sensors in our aquaponics system, and the other project is about brewing our own fertilizer tea from the sludge or the sort of captured fish waste, sediment, which includes fish food, fish waste, maybe even some organic debris, removing it from the system and then making a fertilizer out of it. How do you even go about quantifying that? How do you even go about thinking about that process to know how good your fertilizer is? So I've sort of opened the door to that world, and I'm basically going to bring you in on where I am right now as far as that project, but I'm nowhere near finished. So we'll start with the sensor project. So we, the main parameters that we are testing are, the usual ones: pH, temperature, dissolved oxygen, as well as nitrate, nitrite, and ammonia. Pretty common for aquaponics people. We've also added carbonate hardness, which is another recent breakthrough that we've had in understanding the buffer of how pH changes. So I can even have a separate discussion on KH, because we're really not concerned as much about the fluctuation of pH after we have focused on KH. But for the point of these sensors, I was having inaccuracies with my home DIY API tests, if you've ever used them before. I even did we did some comparisons with our lab here and found that there was up to a 10% error. And I'm also colorblind. And if you're somebody who's reading color-coded tests, I couldn't actually read them myself. I don't know if the students don't know that, so don't tell them. But I would always be like, well, what do you think? And then I would just go with that, But I was really just kind of disappointed and I know it's expensive, and I know it's a thing that takes time to do, and we've done titrations, and we've done all different kinds of water quality testing. And ultimately, if you're a business, you want to spend the least amount of time, but you want to have the most accurate information possible. So I got an engineer to work with me. He was a graduate student from Spain, and he came over and worked for a semester. And we basically found what we think was the cheapest way to measure. And what we ended up measuring was pH, temperature, DO, but then we did nitrate and ammonia. So it's kind of the beginning and the ending part of the nitrification process in an aquaponics system. We thought that if you know ammonia and you know nitrate, you're going to have a pretty solid idea of what nitrite is, even though we are still testing nitrite once a week with another test. So we have two different systems that we have used, and one is called an Arduino system and one is called Vernier. And these are basically companies that produce this hardware, and they have dedicated software systems. Basically, they're called transductors, or they're called the brain of the system. And when you attach a sensor into the water, it's then going into this brain, and it's taking, it's basically sending an electric signal that can then be sent to an IP address where you can download the information in a CSV file. Or one of the coolest things about this, what we've done, is we have the information available on our smartphones. So my students can walk up to the system and scan a barcode that's right next to the system, and all the information for DO, pH, and temperature will come up in graphs that are averaged every 5 minutes. So it's way more data than you'd ever need. But even going forward, we have a system that's connected to Twitter. So it can even send out a tweet from its own system to tell you on your phone that your water quality is out of your set parameters. So, if you're somebody like me who wonders what's going on with water quality when you're at home, you can literally pull up your phone and see dissolved oxygen, see pH, see those changes happening. And there have been times where you see a big drastic jump or a big drop off, and it's correlated to some physical change, like maybe a shut or a float valve has been stuck and there's water just dumping into the system, And otherwise, we wouldn't really know what had happened or when that happened. But now we know we can track these changes by the minute, which is way more data than I would probably ever need. But I wanted to demonstrate that this could be done relatively cheaply. So we designed 2 systems, and when we go around the tour, I'll show you one of the. Actually, I can show you each of the systems. They're about $700 total per system. So you can get the 5 measurements: nitrate, ammonia, pH temperature, dissolved oxygen, for about $700. And you would have to do a little bit of coding, specifically with the vernier system, in order to shape that information into a way that's readable. So there is a little bit of a DIY nature to this, and I had 0 experience with coding or engineering, software engineering, that sort of thing. And not that I had mastered it through this process, but one of the best things is this student came out with a manual that is basically the most dumbed down version of anybody could pick it up and build one of these systems on their own. And at the culmination of this, if you'd like, I could simply just send you the manual by email. So you can even set up more alarm notices. Again, we have the barcode scanner, so it's also sort of a cool piece to where anyone from the public can walk up and they can look at our water quality measurements, and it just helps them maybe understand what aquaponics is and how it functions, and what they can even look at the ranges of our water quality and say, oh, you're out of range, and we'll have to scramble and make something work. But it's a really cool access to information, and if you desire that process or want to maybe try one of these out or just take a look, I'd be happy to share that with you.
Cool. Can you tell me quickly about the sensors? Do you have calibration requirements? Or,
yes.
What's the day-to-day?
Good question.
Keeping it running.
We calibrate the ammonia and nitrate sensors every time we use them. So that process, I don't know, Brendan, would you say it takes about 10, 15 minutes?
Got it down to about that.
Yeah, so nitrate and ammonia, they take a little more time. However, we found that with pH temperature DO, they don't need to be calibrated maybe once every 6 months. And so, did you want to add something to that?
Yeah, I was just going to say that the nitrate and ammonia sensors aren't continuous, so they're not always in the water. So we take water samples and test them twice weekly.
Yeah, we actually have to pull those out. We can't keep them running continuously in the water. So that is also a difference that between these 2 systems, where you can have a continuous running sensors with pH temperature.
That means the ammonia you have to calibrate each time you...
Yes, for the ammonia and the nitrate, but as I said, for the other 3, they don't use much calibration. We found that the margin of error is pretty small. And I have used, I don't know if you've ever run through handheld meters. I've spent thousands of dollars on these portable Hanometers. They last a year, and they're not very accurate. I've used the API tests. Maybe you could argue they're good enough for the information that you need, but I would like to have more accurate information. I would like to make sure that the person doing the recording of the test doesn't have subjectivity, such as me, who is colorblind. Oh yeah, it all looks red.
I read a great, really well-written article about the primary desktop lab kits that Lamont and all those, and they had 4 different people do it, and the range was 20%. Across the board, it was such a range between the way people perceived the tests and the way they did them or didn't do them. It was virtually useless, except for if one person did it all the time, and then at least your error would be consistently bad. But it was disheartening that the data you were getting was only so.
And not only that, Andy, but you may know that you have fluctuations that happen within a day, sometimes within an hour.
It would be really
right
cool
after
to
you
see
feed
that,
the fish.
especially with your nitrogen stuff.
So and we have the data. Anyone can look at the pH temperature DO stuff, and even DO is affected by fish feeding. There's a drop. Yeah,
one thing I would add to that, Kevin mentioned the KH. We've been experimenting with adding buffers, so having continuous pH monitoring allows us to get much more accurate with how we dose something like a carbonate buffer. It's a calcium carbonate buffer. And,
as opposed to the hydrates.
Right.
Yeah.
So
I've been doing that.
sort of dialed that in because we don't want to overdo it. We're able to get very precise measurements of pH.
Yep. Cool. So again, if you are interested, I will send you the user manual and you can eat your heart out. Go for it. The other project that I wanted to talk about was actually not finished, is about brewing and making fertilizer from sludge, or from, I call it sludge, you may call it something else, but really the sort of detritus, waste, solid debris that builds up in aquaponic systems. And you know, a lot of aquaponic systems are different, so you may have different places where it builds up, or different ways that you manage it. But I would argue that it's pretty unanimous that in aquaponic systems there is buildup of debris in some places, and cleaning happens. And what are we doing with that stuff? Are we throwing it away? My sort of thought was, there's something of value in there. There's nitrate in there. Could we use it as a fertilizer? And within the context of this campus, I have a theory that I could be producing enough fertilizer from a small aquaponic system that I could fertilize the entire campus. And that was sort of the crackpot idea where it got started, right? And luckily I work in this place where I can carry out these ideas and actually experiment with them. So I grabbed a student and one of our chemists who helped me identify the process. So basically the question was, what is in this waste? And particularly at the microbial level, is there a way that we can influence the microbial count before it's actually used in a soil environment? So what we did was we looked at different brew times. So we're essentially taking out our sludge waste. We are adding it at a consistent rate. We're dewatering it, adding it at a consistent rate, and then we're feeding molasses as a simple sugar feedstock to basically increase the bacterial population. And then we are measuring basically bacterial total potential using this process called the FDA, or fluorescent diacetate assay process. So again, we kept things, if you've ever brewed compost here, you want to make sure that there's constant oxygen brewed into the solution. And so we maintained above 6 ppm, which is considered an aerobic environment, which is very important because you want to have aerobic bacteria going into a soil environment. Very, very important. Anaerobic bacteria and fungi are tied to a lot of disease and even tied to pest pressures that can happen in outside environments. So our process is basically, and I'm not a chemist, I'm not a biologist, so I'm going to explain the version that I know, and I may not be able to answer your questions, but basically you have a chemical called fluorescent diacetate, which is hydrolyzed by enzymes that exist in these bacteria, right? And when you have a process that happens where you add water to FDA, it basically produces this fluorescent, which can be visualized in the cells, in the microbial cells. So you have basically a color spectrum that's produced that can then be compared to each other using a spectrometer. And you can basically measure a light absorbance, which is giving you a comparative analysis of microbial activity. In order to do that, you have to establish what's called a stock curve comparison. So you have to have known fluorescent concentration and light absorbance, so that when you are getting your final readings from the spectrometer, you then can correlate that to your stock curve to understand what the concentration is in each sample. So we are basically trying to figure out how long do we brew this sludge tea in water in a high oxygen environment to produce the most microbial biomass that could be then put into a soil environment to have the most quantified, beneficial fertility in an outside soil environment. Why are we measuring microbial bacteria? Well, microbial bacteria are very important, and I know we've talked about killing bacteria a lot in this, but they're extremely important to how plants grow in making organic matter converted into a bioavailable form of energy. And in fact, 90% of energy that's produced in soil goes through these microbial decomposers. So understanding your bacteria and your fungi is basically helping you understand your health of the soil and how productive it can be. And that is a little bit different than your standard NPK test, which is telling you just your dissolved nutrient matter, but it doesn't really tell you this bacterial potential, the microbial, the living potential of what's in soil, which is making energy actually available to the plants. So that was the big question, and I got it from the study done in 2005. Now this was adapted. This is done on soil, and so we are doing it on a slightly different process with our sludge. And we're basically in the middle of doing this experiment, and we'll have our results in about a month or so. So I just wanted to explain what I was doing, but also kind of open the air to a discussion to you guys to, and actually the whole part of this project got started was a conversation I had with Ben, who was interested in the idea of selling aquaponics water from a system. How can it be treated? What do we know what's in it? How can we put something on the bottle to say, here's what you're getting? And in many ways, there were just a lot of question marks in this area. So this is kind of where I started, was I see another potential value in the aquaponics business for pulling out fertilizer, for making fertilizer tea. And this is sort of the path that it led me down. And it opened a door to many, many other doors that I would like to continue moving towards. But it's really just kind of the first step in a discussion of how can we provide more value, how can we make more money from our systems, aquaponics or hydroponics. So that's sort of the end of that project. I can try to address any questions if you have on this one, or if you have any suggestions, or if you have been interested in selling aquaponics water. Another thing that I've been concerned about is if we're selling a tea product, that it's losing oxygen relatively quickly in a bottle. So if you produce this really high oxygen tea and then it sits on a shelf for a month and somebody buys it, in my mind that's not the same product than what was bottled. And the difference of having air in the bottle that can potentially feed these microbes versus having the bottle filled to the top could be the difference in how much oxygen is lost and how the microbial life will then change in this solution. So I'm not convinced that we can actually produce a viable product that has a shelf life if we're talking about a living bacteria. And so that's another challenge to this, is how do we make it work in the marketplace? Yeah.
So right now, the microbe products they sell on the shelf, they usually get the microbe to go in some sort of like stasis or hibernation mode. Plant Clinic might know more.
Sure.
Or they add something into it that would stabilize it and stop it from changing. So
interesting.
That's your biggest problem, is keeping things consistent.
Okay. I'll have to look more into that.
Yeah, just Google viable but non-culturable.
Viable but non-culturable. So what is it exposed to when it's...
They don't really dead. They are not dead.
They will grow after some time.
It's like if they're put in a stressful environment, they'll kind of like protect themselves so you can't culture them. They're not growing, but like she said, they're not dead. So eventually they can be brought back to life.
It just
if you will.
take you a longer time.
Does that lend itself to any ideas of how the microbial life could be changing in this bottle? Like one microbe maybe in a stasis state, but if you're losing oxygen in this, you're basically changing the conditions in which they're potentially growing. So I would be concerned about producing new.
Proportion of different bacteria will be different after some time. But like once you expose them to oxygen, they will do their work to really establish themselves to the stable community, but it will take some time.
And it also probably differs depending on the microbe.
Yeah.
So maybe it might be worthwhile to try and isolate one and then just sell that isolate. I don't know. Easier to store. It'd be hard.
Cool. And then the last thing I just wanted to talk about before we go on the tour was, again, I really enjoyed having an event like this because I just don't feel like there's a lot of connections that we're having. I've seen some of your systems. They're very different than mine. Your system is different than his, and so. So I realize that we're not all doing exactly the same thing, but we have a lot of the same challenges. I think some of the benefits in working together could be applying for grants together, could be sharing of resources or materials, could be buying and selling materials, could be helping to support an industry. I don't know, but when Zach and Sarah brought this event together, it was just an idea that I had of how rare is it that we get these people in one room, and it's not just people that are doing it in a closet in their house. It's people who are actually, producing, trying to help the for-profit business, trying to produce research, trying to push this in a way. And I think most of the knowledge in Chicago around hydroponic and aquaponics probably in this room. And so I would love to hear your feedback. What do you think about creating a network, creating a once-a-year meeting? Should we include aquaponics and hydroponics under controlled environment ag? Should we separate them? Should we only have aquaponics people working together and hydro people working together? And if it comes down to it, I would love to be the one to host it. If it's here or whatever, if it includes me putting together a Google group, I'm certainly okay to do that. But many times I just feel like I'm working on my own island. And you might feel the same way too. Or I just think that some of these questions we could hash out and we could communicate over the year about Pythium, about some of the things I've learned today. I would have loved to have been in discussion about it instead of just a one period, one single time thing in which I'm probably going to remember half of what if I'm lucky.
Well, this was really nice get together, and the folks who presented it was really very interesting stuff. So,
are there
if you want to do all that heavy lifting, I'll come. Especially if you feed me, I'll come.
Well, I think part of the challenge is you have to have people to present. Yeah. And my thing is it doesn't have to be research. I think within the field of hydro and aqua, it's a lot of applied knowledge. And it's a lot of like what works for you and how can you make money from it. We don't always have to have the dense research, which it is wonderful to have as part of that because that makes it legitimate, right? But I think we can have all different kinds of points of view, and somebody like Ben who's working in the for-profit model, somebody like you, Andy, who's maybe more of in a teaching model, but also on the high production end. We all have sort of different perspectives and missions, but yeah, I'm open to hear your ideas.
I just think it would be helpful for those of us that came and spoke today, because a lot of what we're doing, whether it be pure research, kind of the hard science, or even, again, like at the clinic, we don't do a lot of research, but we are trying to put together services, knowing what our clientele needs. And part of that is getting grant money, but part of it's also just developing, one thing that I've struggled with is that, as I mentioned before, it's hard to find good recommendations for management in certain systems because they either just don't exist or they're not legally, if it's not legally labeled, I cannot recommend it if it's a chemical. But there are some kind of workarounds with that. But having some sort of knowledge of what it is that the people that are actually putting this into practice need, I think would be really helpful for some of us because, like you said, it's easy to work kind of in a vacuum and you put something together and you say, well, I hope someone finds this useful.
Right.
But knowing, this is a need that needs to be addressed, yeah, we can work on that, or this is something that would help you, sure, we can put something like that together. So I think it'd be helpful from our end.
Is there enough crossover in the aqua hydro worlds that we can work together?
Yeah, so if you can have a Google group, that would be great.
I was thinking the same thing. We could just start a Google group so there's more of an online conversation, and then we just set up a once a year big meeting kind of a thing with food, of course. Right? It's all tilapia and basil.
I don't know, I think we might be a little more cohesive if we met more often.
Yeah, twice a year.
Twice a year?
Every quarter.
Yeah.
Well, here's the concept for the meetings, and I don't know if anyone here has heard of Good Greens. Yeah, they do a sort of a monthly get together, I think, and they have contributors that come in. That's sort of the model that we did here, which I think was fun. And maybe we could have that model, and then also people could submit questions.
Exactly.
Maybe a presentation.
Yeah.
you or a researcher could come in and address the question of practitioners.
I also think we should be seeing other systems.
Yeah.
It'd be great to visit.
We'd be
Yeah.
happy to host, we're
And you
6
would-
months out, but we'd be happy to host when our whole thing, we're moving our whole operations.
Yeah, we're all excited to see you.
Yeah, right.
We're all waiting, Andy.
Andy's waiting there. With 55, I'm gonna have it.
Maybe we can start with a two
Let's-
times a year
Yeah.
meeting. And so what I'll do is maybe I can work with Sarah or Zach to get your email information.
Mhm.
And if you guys want to send out
Yeah.
invitations to people that you know that are really involved, again, I would probably try and steer away from having people involved who are like just starting aquaponics and building a hobby system in their home, or I don't know if it's like the right group for those people, and they can also come out of the woodwork. I get emails from people building aquaponics systems in their homes every week, so I don't know if we want to overwhelm it with that group of people.
Although we could, it wouldn't have to be the next meeting, but at some point we could have a meeting for those people.
For those people, I
I mean,
think
it
you're
wouldn't,
right.
it wouldn't exclude, other people. I'm definitely a beginner too, even though I'm an educator. But we could have a meeting for those people that would focus on that, that type of system or that type of those questions and circumstances.
Sure, sounds good. Is anyone else interested in jumping to join
And if you
forces
can
with me?
advertise to more people, we will be grateful. I'm trying to get people from other states too.
So this is the first meeting that we had planned, but Helen and I are already in discussions about doing this again in the fall possibly. So
Cool.
Good.
whether that's up here or on campus, if you're doing with Loyola, in conjunction with Windy City Harvest new Lawndale site, I think it's a great idea of facilitating something around both research and applied growers as well. So, is there anything you wanted to add?
Yeah,
Helen?
so I think next time we will try to have, like more of you, people like you to present. So this the first time we do is research heavy because that's what we know. We don't know you.
That was really,
or at least
that was
I
really
don't.
nice of you. Don't not do that. The research
Yeah.
stuff today was
It's also
great.
very
It
unique
was really
that
great.
Yeah.
you would find research in this area.
So
on
I
this
think
subject.
that I think having a Google group is great because that will guide us to do the useful research. It's really boring to do research that no one uses. Seriously, it's really boring because all the research we do is really hard work. So we want it to be as useful as possible. Yeah. So I think before the tour, I just like to say a few words. So of course, we would like to thank Zach and Sarah.
Yeah.
Without them, it would not happen because, I know how to manage a research group, but other than that, it's hard to do. I would like to thank Kenvin and Loyola because they got really short notice and they put it together, trying us the location in like 2 days. While we tried months, some other places that I will not name. I would like to thank Diane. They have been working with my group very, very well, and we really appreciate her help. A lot of data we present are collected from farms that the owner are here. You know who you are. So I'm really, grateful to be able to get, the samples that we book you all the time and allow us to do all the research that we present today. I would like to thank my research group for doing all of the hard work. Thank you for coming, and I hope that we will see each other more. And last and not least, I have to thank the funding agency. So this workshop is actually funded by USDA NIFA program and also EPA project specifically focused on human health impact of water reuse and non-conventional irrigation water. And my passion is on a small system urban farm, like Jack knows about that. So thank you everyone for coming.
Thank you.